Okay, this is the second lecture about primary production and phytoplankton in the oceans. We're spending a lot of time on phytoplankton for good reasons. First of all, perhaps the best reason is that if you know what's happening with primary production, the types of phytoplankton, the level of primary production, you can say a lot about what else is present in the system and in the habitat. It really sets the stage for everything else that happens there. So, so let's learn a little bit more about these organisms. Um, you've seen already about how light impacts. Well, we're going to talk more about how light impacts phytoplankton, but you've seen already about how light interacts with the water column and how it decreases exponentially through the water column as a function of depth. And we're going to talk much more about this on Thursday and go through some examples. That's going to be basically the whole session on Thursday will be an in-class exercise at looking at this equation and how light intensity varies as a function of the attenuation coefficient K. And that coefficient varies with wavelength of light, that is the color of light. I hope you remember that that light is attenuated at either end of the spectrum. Both the short and the long wavelengths of light are absorbed more so by water than in the green, in the middle part of the spectrum. It also varies with the type of water in terms of how turbid or clear it is. Basically, more turbid the water is, the higher the K and more the light is attenuated. Here is basically a picture of how we measure light attenuation. Basically, there's a cable that's connected up to the computer, and this red cable is actually what's lowered down, connected up to this part of the equipment down to the water column. This instrument right there, part of the instrument right there, is set on the deck of the ship that measures the surface radiance to give us I sub O. So anyway, that's how we measure light. Really important, obviously, for these photoautotrophic organisms. Just a word about how we measure primary production to understand a graph in a minute that I'm going to show you about how primary production photosynthesis varies with light. Here's our equation. You can say this is the equation of life. If this doesn't occur, we don't exist, basically. And it shows the fixation of CO2 to organic material. Again, that CH2O is not a specific compound, just supposed to represent organic material. The equation also shows a very important waste product from this reaction, oxygen. So we can measure primary production by following basically two elements. One way is measure the production of oxygen. That's fairly easy to do. The only disadvantage of that is then you have to make some assumption about the relationship between carbon dioxide fixation and oxygen production. And usually 1-to-1 is what's assumed, but there are cases when that's not the ratio. Another, in some ways, even more common and easy way of measuring primary production is by following the carbon. And that carbon can be traced with a radioactive tracer, C14, or with a stable isotope tracer, C13. Turns out it's easier to use the radioactive form. The piece of equipment for doing that only costs about $20,000. We have one down here in Lewis. There's others up in Newark, I'm sure. The C13 method, although it's not radioactive, which has some advantages, it's harder to measure, and there are machines around here that do it, but it's much more difficult to do. Anyway, that's how we measure primary production, by tracing either oxygen or the carbon. So the question is, what's the relationship between primary production and light? And light here, I'm going to use the loose term light, but what we really mean is photosynthetically active radiation, or PAR, which is, you can see here, the wavelengths that PAR occurs. So here's a graph showing how primary production, now measured as oxygen, varies as a function of PAR. And just first of all, before we get into the details of the equation, just note the general shape of the curve. Basically, it goes up with light, and then we see this leveling off. And this general shape of the curve is very common in ecology and in marine biology. That is, things tend to go up with the property, and then they tend to level off. We're going to see this in a minute with regard to nutrient uptake. We're going to see it later with regard to grazing, and here we're seeing it with regard to light and the rate of photosynthesis. The equation, there's several equations that are used to describe this curve. This is just one of them. I don't think it's important enough for you to remember, but what is important to remember is the parameters. First, the easiest one to look at is P max. It's just basically the maximum rate of photosynthesis where light is said to saturate out prime production. The other parameters that are often mentioned are alpha, which is basically the initial slope of this relationship. If we would fit a straight line to that relationship, that's alpha. And you can see the relationship between alpha and P max, and that relationship there is called I sub k. So those are important properties of how prime production varies as a function of light intensity, or PAR. Now sometimes you actually see the prime production decrease with really, really high levels of light due to photo inhibition. Now because of that relationship between light and prime production, this is the relationship you see with regard to depth. And so here's light decreasing exponentially with depth. You should know the equation for what's happening with why that's occurring that way. And there's prime production there. And you can see here's that little bit of inhibition of prime production at near the surface of the water column, and then it kind of reaches a maximum and then it starts to decrease. So that's how prime production varies as a function of light and how we see it going through the water column. So now let's talk about the controls of prime production. We've hit all these other questions. You should know already what prime production actually is. That's pretty easy. We talked last Thursday about how light is harvested by prime production, by phytoplankton that is, by the different accessory pigments and to a lesser extent chlorophyll A. You should know already the different types of prime producers in the oceans, the diatoms and cotylophorids and so on. And now we're going to talk about what controls rates of prime production. And there's a really simple answer to this, and of course a much more complicated answer. The simple answer is nutrients and light. And next few lectures we're going to be hearing the more complicated answer to that question about what controls those rates. So one way to get into this and to kind of in some ways test our answer to the question about what controls prime production is to look how it varies with the seasons. And if we can't explain why prime production varies with the seasons, it's saying we're not doing a really good job in explaining what's controlling it. Because if we can't explain it, then, well, we just don't know what's going on then. So the observation that we need to explain is the fact that there's a spring bloom. There's this peak in prime production that's in the spring, in May or April or so. What spring kind of depends on where you are. But so let's just kind of leave it a little bit vague and talk about just the spring. And this problem is of course only really applicable to temperate regions, like where we live, and this band of the globe. Because in the tropics, of course, there's really not that much of a variation in all these parameters, physical parameters that we're going to be talking about today. So what we're really talking about now is what's happening in temperate regions, such as off of our coast. And then off of our coast, there's often, again, this spring bloom. So why does that occur? And basically, part of the answer is given here by this interplay between light and nutrients. So here's a little bit closer to real life, showing the spring bloom off of our coast, the whole east coast of the U.S. A couple points here to note is that here's a use of chlorophyll A. What you're really seeing is the reflection off of the ocean that's due to only chlorophyll A that's detected by satellites. And also note the scale of this. The really a high area, dense biomass of chlorophyll. So to back up, you remember that chlorophyll A is used as an indicator of the biomass of these organisms, just how much phytoplankton is present. And so the real high values would be about 10 or so. And generally, this has milligrams per meter cubed, but generally we'll talk about exactly the same. It's micrograms per liter or so. So 10 is pretty high. Maybe sometimes in the Delaware estuary we get up to 20. So if you just look at this long enough, you'll see these pulses of really high red values and so on that should be occurring right around spring. It's kind of hard to see. You have to kind of change your eyes between the actual map and the clock. But I hope you look at this long enough to see the pattern. Of course, another aspect of this graph or map is showing the variation not only over time but among the different areas along the coast. Prime production obviously varies with locations and different regions along the coast. We're going to be talking a little bit more about how prime production varies spatially around the globe more on Thursday. Today we're going to be focusing on that variation over time. So back to the question, why is there a bloom in spring? And the answer is, well, it's the best combination of both light and nutrients. There's enough of both of those things to allow the phytoplankton to take off. So in terms of nutrients, what we mean by that is nitrogen is the most important limiting element in the oceans, and the forms of nitrogen include nitrate. That's the most there's more nitrate than any other nitrogen form of nitrogen except for nitrogen gas, which these organisms cannot use. Much less ammonium. We're going to talk about the difference between the two when we talk about the grazers. But to give away part of the answer, grazers release ammonium, whereas nitrate comes from the deep waters. So in addition to nitrogen, phosphate is required by these organisms, all organisms, and then only diatoms need silicate. Because anyone remember why only diatoms? Well, it's because diatoms have silicate in their cell walls and their frustrals, whereas all the other phytoplankton do not. There's only a few organisms in the oceans that they require silicate. There's other things that are required. These are the so-called micronutrients. Much less concentrations or less of these nutrients are required by phytoplankton. And we'll talk quite a bit about iron. Well, at least we'll talk a little bit about iron, not quite a bit, on Thursday. So those are the nutrients that are controlling these organisms. So let's get back to why is there a spring bloom? And let's start off the story with the case in winter. Basically in winter, there's no stratification. It's cold, not much light, and there's a lot of wind. And so if we look at the thermocline, that is there's no thermocline, there's no change in temperature down to perhaps 100 meters or even deeper in, say, the North Atlantic Ocean. Now, if we look at so this again shows us basically the same thing. The density of the water is pretty constant for a little bit of unclear how deep, but at least 100 or so meters. Light, well, light is there's not much of it because it's winter. The days are short, and so it's there's not a whole lot of light. Nutrients. What about nutrients? What would those look like? And part of the answer is given right here by the density profile. That is pretty straight up and down. And so yeah, nutrients are also pretty much straight up and down and high because you're getting very deep water that's being mixed up because of the wind with high nutrient concentrations. So now if we go to the spring, what's happening during the spring? Well, spring, the sun comes out. It's starting to warm up. And so we see a increase in the temperature of that surface layer. And what's called this diagram has this phrase thermocline growing, which is a little bit not quite correct. What's growing here is the mixed layer, and what's forming is the thermocline. That is a change in the temperature. Thermocline means change in the temperature. And so that's allowing stratification. That's allowing stratification. And so this, I don't want to emphasize salinity so much, but I want to emphasize the change in the temperature. The warmer temperatures are setting up a less dense layer of water on top of a more dense layer of water. And that's showing up here as lower density of water on top of higher density of water. So that's what's happening in the spring. So what does all this have to do with blooms? And can we use that to predict when the bloom actually occurs? Well, the guy who kind of came up with this idea and first had a credible explanation for why blooms occur was Harold Sverdrup. Sverdrup was actually Norwegian, but came over to this country, and actually I learned just as I was looking up what the heck his first name was the director of the Scripps Institute of Oceanography back from about if I remember correctly, 1936, 39, something like that, to 1948. So basically during World War II. But he's actually much more famous because of not because of being the director of Scripps. By the way, Scripps is perhaps one of the most famous places to do oceanography in the country, that and the Woods Hole Oceanographic Institute on this coast. So anyway, I'm sure many more people know Sverdrup because of his critical depth hypothesis and his other work in physical oceanography than the fact that he was the director of the Scripps Institute of Oceanography. So anyway, he came up with this idea, and he made a couple simplifications and assumptions. Very common to do this. And one of them was that he just said that prime production varies linearly with light. And we know already that's not the case, that there's this sigmoid relationship between prime production and light, but this turns out to be just, back then it was much more important. We could actually easily not have any of these two assumptions and still talk about this idea. But back almost 80 years ago, not 80 years ago, that's some 60 some years ago, and that I was born not too long after 53, so I'm not quite that old. Anyway, back then there just wasn't the computing power to do a lot of the calculations that you will be doing quite easily as part of the homework that is due on Friday. Anyway, so these assumptions made it easier to look at this whole question. So in addition to assuming that prime production follows light linearly, basically more light, more prime production, he also assumed that respiration, that is the loss of carbon, that is going from organic material back to CO2 and the consuming oxygen, is constant with depth. And then he deduced that, and this is the crucial point, he deduced that you would have a bloom when the integrated, this is crucial, integrated prime production exceeds respiration. So let's walk through this a little bit. And this is a diagram actually from that 1953 paper showing again the decrease, the exponential decrease of prime production. That's exactly what light does. And again, here's respiration. It's constant with depth. And he noted two important depths in this relationship. One is the compensation depth. And the compensation depth is when the per meter cubed rate of respiration equals the per meter cubed rate of photosynthesis. So that's right when those two lines cross each other. That's one of the depths that he noted. But I think what's much more important for this whole idea about when do blooms occur is the critical depth. The critical depth is when the integrated, so it's meters squared rate of photosynthesis equals the integrated rate of respiration. So it's not when these two curves cross, but it's when the area here in the respiration box equals the area here in the photosynthesis. It's not a box, it's whatever, that area underneath that curve. And so in this diagram, it's somewhere down here. And this is precisely what we will be exploring, what you will be exploring for the homework. And what we hope to do on Thursday is to provide you with the tools for looking at these equations using Excel to answer this question. So Sverdrup hypothesized that blooms would occur when integrated photosynthesis is greater than integrated respiration. So in other words, when the gains of photosynthesis are greater than the losses due to respiration. And then, so when a bloom occurs when the depth of mixing, so if the mixing is less than the critical depth, and the less is the critical depth. So the depth of mixing is that, so there's water, so these organisms are being mixed up and down because of the winds. If that depth is less than the critical depth, there will be a bloom. There will be a bloom. And so if we look at this diagram right here, the question is will a bloom occur or not? Unfortunately, this is a recording, so I can't see what your answer is, but I hope you're thinking about that. So the answer is no, because what's going to be happening is that these organisms are going to be mixed all the way down here, and so they're below the critical depth. And the area of the losses, the respiration, is greater than the area of the photosynthesis. And so the losses will be greater than the summed up losses will be greater than the summed up rates of primary production or photosynthesis. So there can't be a bloom. So as I said, as I promised, you will be looking at this in more detail as part of the homework, and you'll be learning how to approach this problem on Thursday in terms of learning some tricks with Excel to set you up to do the work. So this critical depth idea has been around for a long time, and as you may guess, it's been really critically reviewed, and some think it should be just totally dumped completely. And there's some good arguments about why it's just plain old wrong. That's what Mark Barenfield has argued in this paper that appeared a few years ago. And so you may ask, well, why in the heck are you teaching us something that's just wrong? Well, even though it may not be totally correct, it has various interesting aspects of it that help us integrate these ideas about the impact of the physics on the biology. And it's a great example, I think, of how those two, physics and biology, come together in terms of helping us explain a big feature of this ecosystem, that is the spring bloom. So although the critical depth hypothesis may not be entirely correct, I believe it has enough truth to it, if you will, that's still very worthwhile looking at and exploring. Okay, so what you should know now is the sigmoid relationship between prime production and light intensity. You should know that, be aware of the fact that at really high light intensities, the phytoplankton can not be so happy, and actually their rate of prime production decreases. You should know that blooms form when phytoplankton have enough of both light and nutrients. Also, another critical aspect of that bloom formation is, at least according to the critical depth hypothesis, is that the water column has to be stable, in the sense that there can't be any deep mixing. If there's deep mixing, then they get mixed down too deep where it's dark and there's not enough light for them to grow. Even though there's more nutrients, it doesn't matter because there's no light. And that's what the critical depth hypothesis is all about. It's really about the relationship between photosynthesis and respiration, about the gains of carbon by photosynthesis versus the loss of that carbon by respiration. Okay, so now let's move on and talk a little about the explaining the different types of phytoplankton that we have in the oceans. And you learned already there's lots of different types. And you probably know already, we've talked a little bit about this, that they don't all just occur happily together. Some are more abundant than others at certain times of the year in certain areas. And so let's start to think a bit about why, what are some of the factors controlling their abundance. And let's start off with a bloom again. And it turns out that the group of phytoplankton that usually dominate spring blooms in coastal waters are diatoms, and more generally speaking, other big cells. And the question, of course, is why. So one answer about the diatoms is that they just do better at low temperatures. So even though the water is getting a bit warmer in the spring in surface waters, it's still cold compared to the summer. So that may be part of it, but I'm gonna focus more on their ability to take up nutrients, which is certainly the case. And that then leads to the question: why are they better at taking up nutrients in the system at this time of the year than other organisms? And so before we answer that question, let's introduce another group of organisms. And those are the ones that occur in the summer when nutrients are low, because the nutrients have been stripped out by the phytoplankton that are dominating the spring bloom, which now you know are diatoms. They take out all the nutrients and drive those concentrations to really low levels. So what type of organisms are abundant there? Well, it turns out they're small, and I think we kind of alluded to this already. They're called the pico phytoplankton. Pico meaning, we'll see in a minute, they're less than 2 microns in size. And they include 2 cyanobacteria groups. These are both cyanobacteria groups, and these are 2 genus names that I want you to remember: Prochlorococcus and Synctococcus. They're both, Prochlorococcus is a little bit smaller than the Synctococcus, but they're roughly, they're both less than actually a micron in size. And finally, the third group of organisms are just small eukaryotes, sometimes called pico phytoplankton or pico eukaryotes. These organisms were discovered by flow cytometry, a really fancy method to look at these organisms. The original flow cytometer was actually developed and is still being used to look at red blood cells and to just count them, basically to figure out how anemic you are or not. And it was applied by Penny Chisholm at MIT, among others, to look at these phytoplankton. And it was through the use of especially flow cytometry that Prochlorococcus was discovered back around 1986. And so that was one reason why Penny Chisholm was elected to the National Academy of Science and has done quite well for herself because of her great work on Prochlorococcus. Anyway, so these small organisms dominate in the summer when nutrients are really low, and more generally speaking, in the open ocean where there are very low nutrient concentrations basically all year round. So the question is why? Well, size matters. So basically, again, there's a whole set of terms that you're going to have to know to talk about size of organisms and size of the plankton. So the organisms that are basically, again, less than 2 microns in size are called picoplankton. You can see the organisms that occur in that size category. Next one up is the nanoplankton, 2 to 20 microns in size. Those include some dinoflagellates, other flagellates that may or may not be involved in photosynthesis, may or may not be involved in grazing. We're going to hear more about those next week. They include some smaller diatoms, ciliates. And then going up in size, we have the microplankton, which include, again, some of the larger phytoplankton. And then finally the mesoplankton, which include, basically these are the larger copepods and other zooplankton that are much bigger. So these terms are important for you to know. They're commonly used in oceanography to describe these different organisms. So you can see that they're not really following taxonomy at all. Some of these involve different kingdoms even, eukaryotes and prokaryotes. But they're very useful for thinking about the relationships among organisms. So why do the small organisms do well in the summer when nutrients are concentrations are really low, and all year round, basically, in oligotrophic environments. I hope you know that word already. Oligotrophic means low nutrients. It's not writing too well. In the open ocean. Well, all these organisms, all plankton, don't need much supporting material. They don't have much cellulose at all. They have high protein. We talked about that already. These really small, the pico plankton and even the nanoplankton are pretty neutral density in terms of they don't really sink at all. They're too small to sink, basically. Another factor is that they are able to reproduce quite rapidly. That's also an advantage. Basically, there's an inverse relationship between size and how fast you can divide and grow. But really, when it comes down to the main story that I want to concentrate on for the rest of this lecture is the fact that these organisms have high surface area to volume ratio, and that helps them take up nutrients at low concentrations. So just and it also helps gas exchange, although that's not quite as crucial for what we're talking about right now. So why do small plankton dominate in nutrient-poor waters? Short answer is they outcompete the large phytoplankton for nutrients when concentrations are really, really low. So why is it good to be small if you want to take up a dissolved compound? Well, it's because you have more surface area compared to your volume. Now, the absolute amount of surface area is bigger for a bigger organism, but that surface area has to do much more work, so to speak, for a larger organism. If we just think about the area and volume of a small thing versus a large thing, there's equations for describing those two properties. And if you just take the ratio of one to the other, you can see that as an organism gets bigger, the surface area to volume ratio goes down. So if we just look at the volume here as a function, just a volume, and then look at this ratio, surface area to volume ratio, it's going to go down quite a bit very quickly as the volume gets bigger. So small organisms have bigger surface area to volume ratio than big organisms. So how does that translate into taking up nutrients? And how do organisms that are small compare to big organisms with regard to taking up nutrients? So the surface area to volume ratio helps to explain it, but really what we need to look at is the actual rates of uptake of nutrients and growth of these organisms as a function of nutrient concentrations. Now, I'm just using the generic term nutrients, and classically the symbol given for that is S for substrate. For the most part, though, we're talking about nitrogen compounds, and in particular nitrate. But we'll keep to just talking about nutrients more generically speaking, and as I mentioned, S is the symbol that's used for substrate for looking at these relationships. So you've probably heard about this in grade school, about this relationship that's you may have seen first for enzyme kinetics, and that's what it was first devised for or used for. But it's been taken, just transplanted without any change to talk about the uptake velocity of a nutrient as a function of substrate concentration. And yes, you do have to remember this equation if you haven't memorized it already for that test in 8th grade. So basically, again, here's our favorite sigmoid type of relationship between a property of an organism and an environmental parameter, in this case, substrate concentration. And here it's based on, again, a Vmax, the maximum uptake velocity, and also now the half saturation constant, which is the velocity, the substrate at which the velocity is half of Vmax. So Km is the substrate, when V equals 1/2 Vmax. And so you can mathematically prove that by just putting in here, instead of S, you put in, no, you put in, you put in here 1/2 of Vmax, and you'll prove that the S will be equal to Km. So that's one way to kind of, that's one way to derive what Km means, which we won't go through any more detail. But you do have to know that Km is equal to the substrate concentration when the velocity is 1/2 of Vmax. And that's kind of illustrated here. So now Vmax is I'm just going to switch over to the other one, which is cleaner. Oh, this is given up right there already. So the Monod equation is exactly the same. The only difference is that instead of uptake velocity, it's growth rate. And so, I'm going to show some examples in a minute with growth rate, because ultimately that's what matters. And there's going to be a good relationship between uptake velocity and growth rate for a limiting nutrient. So those organisms that are taking up a substrate faster are going to be able to grow faster. So that's why there's good reason to focus on growth rate. So what I was going to say is that if we look at this exactly the same graph, even if the Vmax is 1, we can see here that the substrate concentration at which the growth rate in this case is 1/2 of Vmax or Umax is the K sub m, and that's what's illustrated right here. So if we didn't know that, we could go here to 0.5, go back to the curve, and see where it intersects in there and drop down, and that would be Km. Same equation, but now we're talking here about growth as opposed to uptake concentration. So, well, what's the big deal of that? Why am I being so mean and asking you to remember that equation? Well, it's important because we can describe these different organisms by two relatively simple properties. That is their maximum uptake velocity or growth rate, and this half saturation constant, K sub m. And so if we know what the substrate concentration is, or the nutrient concentration, again, nutrients here are being represented by S, we can predict what's going to happen. Assuming that nutrients are really the only thing that matter here. And again, we're still kind of thinking only about a fairly simple case of what's controlling prior production. We're thinking only about nutrients from now. Well, there's no other organisms yet in our ocean except for the phytoplankton. So in theory, we could predict what's going to happen with a competition between these organisms. And we can predict which organisms should occur where by looking at just nutrient concentrations and knowing something about these parameters, maximum uptake velocity or growth rate, and the half saturation constant. So let's just look at a couple examples. Who should win in this case? What is that, blue or black, or I don't know my colors. Is that pink? Which one should win? Well, obviously the one that's higher up. And so the black one here, or dark blue, will certainly win here in this case, where the nutrient concentrations are relatively low, and it has a lower half saturation constant. So it's going to win pretty much all here. And then only when the concentrations get really high does that advantage of the low Km have any impact. And after a while, it doesn't have an impact, and the relationship is governed only by the maximum uptake velocity or maximum growth rate. So in the end, you can't say who's going to win all the way over here, but here you can see who's going to win. Okay, here's another case. Now, in this case, these organisms have the same half saturation constant, but they have different maximum growth rates or maximum uptake rates. And of course, this guy again, the dark blue, black guy will win over the whatever that color is, pink, magenta maybe, at basically all these concentrations. Now another way to kind of phrase and to kind of test your knowledge of what's going on here is which one is a small cell and which one is a big one. Can you guess? Well, you remember how one way to think about this is we were saying that the small cells should win out when nutrient concentrations are really low. So that one would be right over here, right? here's the big one where nutrient concentrations are high. So that guy's not going to win. Circle the wrong one. That one is going to win when the nutrient concentrations are high. So the winners during the spring bloom are, again, the diatoms, and more generally speaking, the large cells with high or large Vmax or Mumax, maximum growth rate. And the winners in the spring bloom, after the spring bloom, when nutrients are low, and more generally the lake-trophic oceans, well, often oceanographers will talk only about KM, and we don't need to go into more detail about this, but it's really based on the ratio of having the maximum uptake velocity or maximum growth rate and the half saturation constant. So you can see that if this is really low, or if that's really, really high, these organisms are going to win out. Simple answer is yes, a very low KM will help and win, but the really better answer is the ratio is the best predictor who's going to win. Okay, so that goes a long way. The nutrients go a long way to help us understand what's happening with the global distribution of prior production. And this is a contour map taken, I think, from Parsons and Lawley, one of the textbooks that I recommend that you take a look at that's online. Pretty classic picture, for most part based on satellite pictures of chlorophyll A. And from that, we talked about chlorophyll A being a measure of biomass, but the people who do satellite oceanography basically rely on that also to make guesses about the rates of prior production through some magic that we don't need to get into. Anyway, the point here is that you can see from these satellite data that the real high, bright, hot colors are found near coasts. And so we'll talk more about why that is on Thursday, or a lecture that I will record for Thursday. I'm not sure how much time we'll have to discuss it, but I think you may know the answer already. That's because of upwelling that's bringing up nutrients from the deep waters. And some classic examples are off the coast of Chile and Peru, off of South America. So, but anyway, for today, what's important to know is that supply of nutrient explains, is the nutrients are explaining that the distribution of prior production where phytoplankton are most abundant. So the open ocean are basically really the depauperate of phytoplankton, not much there, and much lower rates of prior production, and that'd be equivalent to a desert. And then we have upwellings, which I just mentioned, have high rates of prior production, more equivalent to rainforest. Coral reefs are the kind of the exceptional place. We've kind of alluded to this already. That they are in waters of really low nutrient concentration, so it kind of doesn't follow this pattern at all. And we'll talk later about why they can be so productive. So the units here are given in grams of carbon per meter square per year. More common units are, as you can see there, milligrams of carbon per meter square per day, or sometimes millimoles of carbon per meter square per day. So if we look again about, so those are all just to back up, all per meter squared. And so this is just another diagram showing the same thing. Again, here we have the meters squared for different regions of the oceans. And we can see here again the high rates per meter squared near the continents, the shelves, both upwelling and not upwelling. Coral reefs, again, very high. Seabeds, really high. Those are, of course, near land where waters are shallow enough to have these seabeds. Marshes are really productive. We'll talk more about marshes at the end of the course. On a per meter squared basis, these are much less production going on. But because of the area, especially the open oceans, there's much more prime production going on in the open ocean compared to um, uh, the shelf and upland regions. So it's really important to keep in mind the units, and I'll have a slide about that in a minute. But so per meter square, it's high in coastal waters, but the total prime production in the oceans is dominated by the open oceans, far from land. Also, the other point is that the phytoplankton biomass is low in the oceans, but growth rate, generally speaking, all oceans is low compared to what we see on land. But the growth rate is high. The growth rate is high. And so it's really important for you to keep in mind the units. And so you have these units of per meter cubed, the volumetric measure, versus per meter squared. And that becomes crucial for understanding what's happening with the critical depth hypothesis, and you're going to have to deal with that when you do the homework. Another unit is really a per capita unit, growth rate or doubling time, which has units of days or per day. And so we'll talk about other properties that are best described by those units. So here again, let's I think I mentioned this already about the difference in biomass. So there's much less biomass per meter squared in the oceans as compared to what's on land. But remember that the amount of prime production totally in the oceans is roughly equal. That's if you add up all the prime production done in the oceans, it's roughly equal to what's on land, in spite of the huge difference in biomass. And so the implication is that the turnover time, or how fast these organisms are growing as a per capita, is much, much, much faster in the oceans and, more generally speaking, in all aquatic environments as compared to what's happening on land. So this is turnover time. So this is basically how fast things go through that pool of carbon in terms of the organisms. So this is a small number, but if we would have a growth rate, it would be a very high number. They're growing really, really fast compared to what's happening on land. They're growing on the order of a day in the oceans as compared to years on land. So again, the per capita changes are really important to keep in mind. So this is the same thing that we had already. Turnover time, these are all related. This is generation time. Basically is much shorter. These organisms are not lasting as long. They're phytoplankton for the most part live only a day, as opposed to as much as centuries for a tree on land. The inverse of that is another way of describing that, growth rate or doubling time.